Hello and welcome to Module CCS11 on Contamination Risk Minimisation. This session will discuss risk minimisation by applying best practices in the design and validation of cleanrooms. In today's session, we will discuss the measures implemented to minimise contamination risks, such as cleanroom design, airflow considerations, HVAC systems, and alternative measures to maintain ISO grades. Cleanrooms are specialized environments designed to maintain controlled levels of particulates and specific environmental conditions. Design considerations include managing particles, microbial, electrostatic discharge, molecular and gaseous contamination. Effective cleanroom design addresses critical factors such as airflow patterns, pressurization, sound, vibration, safety, and process impact. The goal is to establish effective contamination control while optimizing reliability, productivity, and cost, ensuring that cleanroom environments support high-quality production and research processes. Ensuring compliance with CGMP regulations for building and facilities and drug manufacturing is crucial for product safety and efficacy. Drug manufacturing buildings must be suitable in size, construction, and location. The layout should allow thorough cleaning, regular maintenance without disrupting production, and support efficient workflows to prevent contamination and maximize productivity. The facility's design should prevent contamination in the flow of components, containers, closures, labels, materials, and products. A seamless, contamination-free transition between production stages is essential. Operation should occur in designated areas of adequate size to prevent mix-ups and contamination. Clear demarcation and adequate space help maintain order and cleanliness, reducing the risk of mix-ups and contamination. Functional Requirement Specifications, or FRS, critical parameters and acceptance criteria define the requirements and performance standards of cleanroom suites. These specifications ensure the cleanroom supports the intended process and maintains a controlled environment. Critical parameters like temperature, humidity, pressurization, air change rates, and particulate microbial counts must be clearly defined and closely monitored to prevent contamination and ensure product quality. Validating cleanroom suites involves several stages, including design qualifications, or DQ, installation qualification, or IQ, operational qualification, or OQ, and performance qualification, PQ. These stages confirm the cleanroom suitability for its intended use and ensure it meets its regulatory standards. Establishing protocols with clear acceptance criteria for environmental factors is crucial. Regular monitoring and adherence to these protocols prevent deviations that could compromise the cleanroom's conditions. Approval by discipline experts and adherence to quality policies are essential for developing and implementing these protocols. This collaborative approach maintains the reliability and integrity of the cleanroom, ensuring it operates at the highest standards of cleanliness and efficiency. Critical zone airstreams for filling and essential activities must meet ISO Class 5 standards. This ensures no microbial contaminants are present, protecting product integrity and patient safety. Surrounding areas should achieve ISO Class 7 cleanliness to support the critical zone conditions and reduce contamination risks. If the critical zone is within an isolator, the external area may be classified as ISO Class 8, suitable for less critical processes while maintaining a controlled environment. Less critical areas are generally classified as ISO Class 8. These areas do not require the same stringent controls, but still need to maintain high standards of cleanliness to support the facility's overall contamination control strategy. The FDA recommends 20 air changes per hour for ISO Class 8 rooms to maintain the required cleanliness. Higher air change rates are necessary for ISO Class 5 and ISO Class 7 areas. Increased air changes in these critical zones ensure quick dilution and removal of any introduced particles or contaminants. CGMP Grade A environments are equivalent to ISO Class 5 standards for both at-rest and in-operation conditions. These environments must maintain extremely low levels of particulate contamination to ensure the highest level of cleanliness for high-risk pharmaceutical operations. The required cleanliness level is typically achieved using a laminar flow cabinet or laminar airflow hood, which provides a continuous flow of filtered air to remove the contaminants and protect critical processes. In Grade A environments, the number of airborne particles smaller than 0.5 micrometers must not exceed 3,520 particles per cubic meter of air. Grade A environments are dedicated to high-risk operations where maintaining sterility is critical. These operations include fill-finish processes in the filling zone, handling stopper bowls, and managing open ampules and vials. Each of these activities involves direct contact with products or components that must remain sterile to prevent contamination and ensure patient safety. Grade B environments adhere to ISO 7 standards during operation and ISO 5 standards at rest. These environments are meticulously controlled to limit particulate contamination, crucial for maintaining stringent cleanliness in pharmaceutical operations. During rest periods, the air must contain no more than 3,520 particles per cubic meter, rising to a maximum of 352,000 particles per cubic meter during operation. Equipped with robust particle monitoring system featuring alarms, these environments ensure prompt response to any deviations. They are essential for aseptic preparation and filling processes, where maintaining sterile conditions is paramount to prevent contamination of critical pharmaceutical products. Grade B is required as a background to Grade A environments when the working area is open to the environment, as is the case with biosafety cabinets. Isolators can be operated in a lower grade background based on risk assessments. Grade C environments are designed to meet ISO 7 standards at rest and ISO 8 standards in operation. These controlled environments maintain strict cleanliness levels, crucial for less stringent steps in sterile production manufacturing. During rest periods, the air quality must not exceed 352,000 particles per cubic meter, while during operation the limit increases to 3,520,000 particles per cubic meter. These cleanroom spaces are utilized for non-critical processes such as preparation area for sterilization, autoclaving, preparation of solutions requiring filtration and weighing, and preparation components or products. Grade D environments align with ISO standard 8 while at rest. During operation, these environments are categorized as controlled non-classified areas where particulate classification is not predefined. This designation makes Grade D environments the least stringent within CGMP requirements for sterile products and may serve as background zone. Grade D environments are primarily used to perform less critical steps in the manufacturing of sterile drugs. These activities include cleaning equipment, assembling closed and sterilized single-use systems using aseptic connectors, and handling clean components, equipment, and accessories prior to sterilization. Thank you for watching this introduction to this module. If you'd like to watch the remainder and get 25% off our Contamination Control Fundamental program, head over to our website at pharmalliance.ie/training-programs and use code YT25OFF. Please do it now as spaces are limited and it's expiring soon. Thank you for your time, and we'll see you on the next module.